Good evening, everyone. My name is Candace, and I'm an event manager at Town Hall Seattle. On behalf of the rest of the staff at Town Hall and the University of Washington, it's my pleasure to welcome you to tonight's live stream presentation featuring cutting-edge research by UW grad students Christine Baker and Hannah Lewis. This is the first of 6 in our series, UW Engage Science, and we're so pleased to be able to offer this platform to these students and their important and engaging work. As we get underway, I would like to acknowledge that our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continued use of the natural resources of their ancestral homeland. Thank you all for tuning in. Town Hall is proud to be a community-focused organization and a place where we can share and sustain ideas and creativity, even if it means we can't gather in person. I'd like to thank tonight's speakers for appearing to help make that possible. If you share in Town Hall's vision for a robust community engaged in the arts, science, and culture where everyone has a voice, please consider donating tonight or becoming a member. Town Hall is adding new programs every day. You can check out more of what's upcoming by visiting our online calendar at townhallseattle.org. Tonight's program will include 2 separate presentations with a time for questions at the end. Each presentation is about 20 minutes long and has visuals with some animation. Questions will be selected from those in the chat field at the bottom of the video player, so please submit those at any time. You can also text questions to 206-504-2857 as noted in the chat. If you can be sure to address the question to the presenter you intend it for, that would be greatly appreciated. We can't guarantee that we'll get to every question, but we will try to get to as many as possible. For those who would like to view the program with closed captioning, click the CC button in the bottom right corner of the video player. The program will also be available for rewatching immediately following the event. Town Hall's work is made possible through your support and the support of our sponsors. Our Arnold Matulski Science Series is supported by Microsoft, KUOW, the Northcliffe Foundation, Wingcoat Foundation Northwest, and the taxpayers of Washington. And finally, Town Hall is a member-supported organization, so I want to thank all the members watching tonight. And now I'd like to introduce our first speaker. Christine Baker is a graduate student in civil and environmental engineering at the University of Washington. She studies the movement of water by currents that are driven by breaking waves along the coast, specifically rip currents, which are hazardous for recreational swimmers. Using large-scale laboratory experiments, she seeks to understand when and how pollutants and unsuspecting swimmers move from the region where waves break on the open ocean. Christine is a native of the Pacific Northwest and grew up in Renton. Please join me in welcoming Christine Baker.
All right, thank you for the introduction. Growing up in the Pacific Northwest, I loved the beach. I could spend hours digging in the sand. Even on chilly days, which were common, I loved to explore with the company of my favorite toys. And most of all, I could spend hours playing chase with the waves. But there are some things that I wasn't quite so sure of. I was afraid of the marine life that I couldn't see below the waves. The first time that my parents took me out to snorkel, I lasted about 30 seconds till I saw a fish, and then I darted on shore. And as I grew older, I realized that, fish, maybe those weren't so scary, but sharks, oh yes, sharks. That was the most scary thing when I swam at a beach. However, now that I spent the last 4 years studying waves and currents along the coast, I'd tell you that the thing that concerns me the most when swimming at the beach has changed. I'm most concerned about bacteria, pollutants, and viruses in the water and the nearshore currents that move them. The nearshore is the regional water from the edge of the beach to about a mile offshore, or about a third of the distance to the horizon. We rely on this region for many reasons. It's a major source of recreation, such as swimming and boating. We move tons of goods to the nearshore. Additionally, we patrol this region for the safety of our country. As many Seattleites might take for granted, we harvest delicious seafood from its waters. And this region has sensitive ecosystems that are home to a variety of unique plants and animals. However, the nearshore water quality is declining due to wastewater and stormwater runoff, pollutants, bacteria, and viruses. Bacteria and viruses can cause human health problems, leading to beach and shellfish bed closures. Wastewater, stormwater, and agricultural runoff can lead to harmful algal blooms, which kill fish and other marine life that we rely on for food. And although we're attempting to clean our coastal waters, there's still loads of pollutants destructing ecosystems. In the nearshore, currents can dictate where pollutants, bacteria, and viruses move. Currents act as a conveyor belt, transporting pollutants and bacteria in the nearshore. And they can dictate if bacteria will sicken swimmers or if pollutants cause destruction of marine ecosystems. I study a specific type of current using laboratory experiments. Before I get into detail about these lab experiments, let's first talk a bit about nearshore currents. To think about nearshore currents, I'd like you to imagine yourself on a beach on a deserted island. In desperation to find help, you write a message in a bottle with your location and you launch it out towards the ocean. Depending on how the waves are breaking, the bottle may remain stuck in one area, bobbing in circles, or maybe you'd see it move down along the beach, being moved by a current. But will it ever move offshore where a rescuer can find it? Where the waves start to break often acts as an invisible fence. Inside the area where waves are breaking, your bottle would move around with different currents, remaining trapped close to the coast. However, there are very few ways that the bottle would move from inside the fence to offshore. One of the main ways is by a rip current, which is a fast-moving jet of water directed offshore, which is what my work focuses on. A rip current might be able to take your bottle offshore, but how do you know when will form and if it will be strong enough to carry your bottle out to the open ocean? Now, more realistically, think of the bottle as pollutants from runoff, bacteria, or viruses. Are there pollutants remaining along the coast near you, maybe sickening swimmers and causing beach closures? For example, the Tijuana River exits into the ocean near the border of California and Mexico. This river commonly carries a lot of pollutants to the coast. The currents in this region can transport the pollutants along the coast, causing the city of San Diego to close beaches or put out swimming advisories. In the map shown here, the red symbols show beach closures or advisories, while the green symbols are open beaches. On last Saturday, there were closures or swimming advisories reaching from the border where the Tijuana River outlet is to La Jolla, a popular surfing location. Beach closures similar to this one in San Diego can occur in Washington state when there's pollutants or even sewage released into the water. Now, are the pollutants instead moving down the coast to a neighboring community? killing fish or leading to shellfish bed closures, as commonly occurs in Washington, on the Washington coast. Year-long closures of Washington razor clam beaches affect over 300 full-time jobs and cause coastal communities to lose about 11 million in income. Lastly, are the pollutants instead moving offshore in a rip current, as shown by this mushroom-like cloud heading offshore, shown with the yellow arrow. This is an image from an experiment where they released biodegradable dye near the shore and tracked it to estimate how much of it stayed near the coast versus went offshore. And this dye could represent bacteria in the near shore. Now, what if the bottle was instead a human? Rip currents pose huge hazard to recreational swimmers. There are over 100 deaths related to rip currents in the US each year. Many factors can impact how hazardous the rip currents are, including the current speed, the size, and how often they occur. These factors can be influenced by how the rip current forms. My goal is to improve predictions and better understand the formation of rip currents, which is complicated by the fact that there's multiple types of rip currents. I'm going to introduce two main types, a channel rip current and a flash rip current. The first type, with which people are more commonly familiar with, is a channel rip current. To think about this type of rip current, I'd like you to once again think about that invisible fence acting as a barrier between the region where waves are breaking and offshore. You can think of the breaking region as a fence, which is holding in a herd of sheep. If the fence were to open at the center, you'd expect that the sheep would exit the breaking region through the fence to seek the green grass in the pastures beyond. Since the gate was opened at the center here, it's not surprising that the sheep exited through it to frolic towards the delicious meal of greens. You can think of this as similar to a channel rip current, or another name for them is a bathymetric rip current, where bathymetry, it means is the shape of the seafloor, which could include a sandbar where waves are breaking on. Like a river flowing through a channel, if the sandbar has a channel or depression, a channel rip current may form there. The way the waves break over the bar, but not in the channel, can cause currents along the beach that come together near the channel and can be forced offshore, as shown by the arrows here. Due to the reliance on the shape of the seafloor, these rip currents happen at predictable locations, just like you knew where the sheep would exit based off the opening in the fence. In fact, on some beaches, lifeguards will know where these persistent channel rip currents occur and keep an eye out for swimmers near them. Additionally, surfers can use these known rip currents to get offshore with less effort. Since they're easier to predict, they're easier to study in the field. There's been many field experiments where people have put out instruments to measure the speed of these rip currents, and even design their own rip current by hammering a barge, which is a giant boat, into the coastline to make a channel. Here's an image of an experiment where they released a green dye to see how fast it moved the dye offshore and get an estimate of the current speed. You could think of this dye as pollutants that's being taken offshore, or even a human. We can use these findings about how rip currents form to make hazard forecasts. For example, NOAA, the National Oceanic and Atmospheric Administration, and the National Weather Service produces forecasts of rip currents. Here's an example of the forecast for the Outer Banks of North Carolina, where the region in red is indicating that they're high hazard of rip currents, while the gray implies that they're low hazards. These forecasts can help places decide when to put out advisories to swimmers and help individuals make educated decisions on when to recreate. The forecasts were developed and tested in areas where channel rip currents are common, and they have shown that these hazard guidance works well in these areas, such as the Outer Banks. However, there's another type of rip current that the forecast may not work so well for. And that is a flash rip current, which is the type that I focus on. To explain flash rip currents, I'll go back to the same setting of the sheep in the fence. This time, the fence will remain closed. Now imagine that some sheep become a little too excited about the prospect of delicious greens that they launch themselves over the fence. You see one, maybe two of them jump over the fence and leap off gleefully into the green fields. But how would you have known that this was going to happen? Is there something about the green grass that could have told you that the sheep would be so ambitious as to jump over the fence to get it? This is like a flash rip current, which are hard to predict and almost seem random. They're also called transient rip currents, where transient means lasting only a short time. What complicates them further is that they do not depend on the shape of the sea floor to form. So there's no need for a channel in a sandbar, for example. Although these events can last shorter times than channel rip currents, they can be hazardous, especially because we don't know where to expect them on beaches. However, we do know that these rip currents do form during certain types of waves. To explain these types of waves that may lead to flash rip currents, I'll start by giving a brief introduction of some wave terminology. So here's a very simple drawing of a wave. The crest is at the top of the wave, while the trough is the lowest section of the wave. The difference between the wave crest and the trough is the wave height, and each wave has a duration, or a time it takes to pass by. Here's an example of what these look like at a beach. As waves are breaking, the crest curls over, or typically curls over, and creates this white foam or bubbles at the surface. So the white crests appear as these bright white sections in the left image, whereas the wave troughs are the lower elevation between the crests and typically appear less foamy. I'll be talking a lot about the length of the wave crest, which is the distance along the beach where you see this white foamy section when the waves break. The length of the wave crests can vary dramatically. When large storms occur far offshore, for example, in the middle of the Pacific Ocean, they create large waves. These waves, known as swell, can travel across the ocean. Swell will arrive at the coast and break with these very long crests. They're common on the West Coast, where you see waves that break along the shore at the exact same time. Surfers love these waves as they make for a wonderful ride on shore. In contrast to that, you can have waves with very short crests. These waves can form during local storms when waves are going in multiple directions. For example, during a storm near the coast of Washington, the winds will cause very choppy seas that lead to these waves that break at the coast with short crests. The waves with short crests are thought to lead to flash rip current events, such as the one you see here. Although we know that the waves with short crests may cause mixing, which can lead to flash rip currents, it's still very hard to predict where the rip currents will occur, so studying them in the field is very difficult. And the rip current hazard forecasts don't reflect conditions that lead to flash rip currents. Because of this, these forecasts may not work well in areas where these type of rip currents are common, such as the coast of Southern California. To better understand how flash rip currents form, I study them in an environment where we can control and make waves with short crests. So for my PhD, I used data from 2 months of laboratory experiments at Oregon State University's Hinsdale Wave Research Lab, which is pictured in this image. Here's an image of the tank. The beach is near the front, and the wave maker is near the back behind the silver bridge. The tank is full of fresh water, and it's the size of an Olympic swimming pool. So it's about 80 feet wide, 160 feet long, and 6 feet deep. I put in a picture of my research team for scale. You can compare the size of the light in these two images. The work that I'm showing today is due to all their hard work collecting these measurements. So one of the things we did was we designed a concrete sandbar, which we put into the tank to make waves break farther offshore. The image on the left is my advisors working hard to put in a section of the concrete bar, and the image on the right is the completed product, where you see that this bar going across the tank with the same shape. So there's no channel in the sandbar, for example. So just like a sheep jumping over the invisible fence, in these experiments, we were actually able to make flash rip currents, as I'm showing with the arrow here. The controlled environment allowed us to test how often these rip currents happened and measure their speed, because we were able to produce specific wave types. For example, in the tank, we can create waves with long crests that represent swell, and contrast them with waves with very short crests that lead to flash rip currents. So the crest length is important. You can visually see that they're different in these images, but we want to actually be able to measure their length and the height of each wave. So how can we do that? To do this, I use a technique called stereo reconstruction. For this technique, we mounted 3 cameras to the ceiling of the tank. These cameras were taking pictures at the exact same time, just from slightly different angles, as shown in the images above. So I can match foam on the water surface between the images and use that information to create maps of the sea surface or the wave surface in 3D. And so I'm going to show you an example of a video of the stereo reconstruction here, which is the plot in the bottom. The color represents the wave height, with the wave crest in red and the wave trough in blue. The reconstruction is in the same orientation as the images above, where the beach is at the bottom and offshore is at the top. You can see that these waves with short crests are coming in, they're breaking at the bar that we designed and running up the concrete beach. From the stereo reconstructions, we can estimate the height, crest length, and duration of each wave to get a complete picture of the wave conditions. Next, we want to see how these wave conditions change the speed of the rip currents, which can change how hazardous they might be. Here's an image of the tank before we made any changes for the experiment. To measure the speed of the currents, we could put out sensors at different locations in the tank. However, this is pretty sparse information, and we'd like to know the speed of the currents in more detail. To do this, we could put out a ton of the instruments in the water. However, this is very expensive and time intensive. So one technique that people commonly use is tracking foam or bubbles on the surface over time and estimating the speed based on how fast the foam is moving. However, unlike the field experiment, the lab does not have very much foam to do this technique. The foam created during wave breaking in the freshwater tank doesn't stick around for very long. So for our experiments, we put in a bunch of soap that cause the bubbles to stick around for longer, more like the field conditions. And we actually added over 8,000 wiffle balls. The wiffle balls were super helpful because they're easily visible in the tank. So you can see a bunch of them going offshore in this rip current here. And you can think about the wiffle balls as representing sheep or pollutants in the water. To estimate the speed of the currents, we can track the wiffle balls or foam as they are moved offshore. If we see where they move over time, you can get an estimate of the speed and direction of the currents in the tank. We can do this for many foam bits or wiffle balls to get a very detailed measurement of the currents in the tank. Results from these laboratory experiments can help us better understand how these wave conditions lead to flash rip currents, which can help us improve rip current predictions. So doing experiments in the laboratory setting allowed us to experiment with a series of a bunch of different types of waves. During the experiments, we could create 45 minutes of one type of wave and then change to another type of wave for the next 45 minutes, and so on and so forth. I showed you an example of waves with very short crests, but we also had experiments where we changed the wave height, the length of the crest, and the water level, and the timing of individual waves. Then we can look at how these conditions impacted the number of rip currents that occurred and the speed of them. So we can ask questions like, when the waves are larger, are the rip currents faster? Or how short do the wave crests need to be to see rip currents? Although my analysis is so far is still in the beginning stages, for now I can say that we can see that there's more fast-moving rip currents when the waves are larger and have shorter crests. We can use this information to improve rip current hazard forecasts. By better incorporating the waves that lead to flash rip currents, we can hopefully improve hazard forecasts in areas where flash rip currents are more common. Additionally, we can use the findings from lab experiments about the number of rip currents and the speed to make estimates of how much pollutants would move offshore versus stay near the coastline, which has large implications for the destruction of coastal ecosystems versus the impact to marine life offshore. Lastly, I'll end by saying that although rip currents are very concerning, being educated about how to identify them, if there are advisories, and what to do if you're caught in them will help your safety when swimming at the beach. Stay in tune with hazard forecasts, such as the NOAA forecast I discussed during the presentation, and watch for signs at beaches about rip currents. Additionally, stay informed about your local advisories. For example, the map I showed you of Southern California was from a nonprofit called Coastkeeper that keeps up-to-date information about swimming advisories due to bacteria levels in the water. Also, Washington State has interactive websites that show salt and freshwater swimming closures, as well as shellfish beach closures. For example, here's a screenshot showing that there's a shellfish bed closure in the Puget Sound, just outside of Seattle, due to harmful levels of toxins in the water. And with that, thank you for listening today, and I look forward to questions after Hannah's talk.
All right, thanks so much, Christine. So our next speaker is Hannah Lewis. Hannah is fascinated by how viruses hijack our cells to cause disease. She's a molecular biology PhD student at the University of Washington studying how herpes simplex virus 1 manipulates proteins that package human DNA. She will explore how herpes makes a home in the nucleus of our cells, where it steals proteins to replicate or to hide from our immune system. Hannah hails from Colorado and is a huge drama nerd, which you will see reflected in her presentation. Please join me in welcoming Hannah Lewis.
Hi there. Thanks everyone for being here. I'm really excited to be here. Thanks so much, Candace, for the great introduction. Yes, I hope you can appreciate that I'm trying to give a flair for the drama for my research. So growing up in Colorado, I always went to the Colorado Shakespeare Festival over the summer. So in particular to drama, I love Shakespeare. So I thought I would introduce my research in the method of a Shakespeare play that you might be familiar with. It starts like this. Two houses, both alike in dignity. This, of course, is the famous first line of Romeo and Juliet, which sets the scene of two feuding houses whose nemesis leads to tragedy at the end of the play. I thought it was very relevant to my research, and I'll tell you why. So let me first introduce the first house that I'm going to talk about. This is the House of Humanity. You're probably very familiar with it. We're all a part of this House of Humanity. And we have an ancient rival, a nemesis. It's a virus that's both feared and hated all over the world. And the virus's name is herpes simplex virus 1. So what happens when herpes simplex virus 1 and humanity come into contact? Well, very frequently, the infection of humans with herpes simplex virus 1 leads to these sores around the mouth called cold sores. They're painful and pus-filled, and they're generally not very fun to have. And this title here says that this is an ancient grudge, and by ancient, I mean quite truly ancient. So 6 million years ago, a chimp-like ancestor of ours was infected with an ancestor of herpes simplex virus 1. And together, we and the virus have evolved over time and to the point where we're now humans and the virus is herpes simplex virus 1. You may be familiar with a cousin of herpes simplex virus 1. About 1.6 million years ago, a similar virus to this original virus infected our ancestors and again co-evolved with us, and this is now known as herpes simplex virus 2. And together, these two viruses have been highly successful. So out of 7.8 billion people in the world, herpes simplex virus 1 infects 3.2 billion. It typically infects around the mouth region, and again, it causes those painful sores. Herpes simplex virus 2 has been a little bit less successful, although I would argue still quite good at its job. And it causes sores typically around the genital regions. However, this is not a hard and fast rule. Herpes simplex virus 1 can also infect the genital regions, and herpes simplex virus 2 can infect the mouth region. And together, although they cause disease in so many people, this disease is typically limited to these sores, and while they're painful, they're not usually dangerous. However, these two viruses can sometimes lead to more severe disease, and examples of this can be blindness, and it can be brain swelling. This happens in about 4,000 people per year. And this severe disease can even be fatal, although this is rare, leading to about 4 deaths per year. I wanted to put in a PSA quickly. Herpes simplex viruses are particularly dangerous to babies, so please don't kiss babies if you have a cold sore. Wait until it clears up to kiss babies. So let's zoom in back to the goals of our players. So again, I'm going to be focusing on herpes simplex virus 1. What are the goals of this virus? Well, it wants to reproduce, spread all over the world. It wants to stay alive, with the caveat that no virus is really alive unless it's infecting a host. And does it want to cause disease? Well, it doesn't, unless causing disease helps it achieve these first 2 goals. These goals actually look pretty similar to the goals of the House of Humanity. We, as a species, would like to reproduce, spread all over the world. We also would like to stay alive, and we'd like to stay healthy. The similarities don't end there. So as a molecular biologist, I study life on the order of very, very tiny molecules, which are the building blocks of life. So I've shown you this diagram several times in the crest of the House of Herpes, and I wanted to go into it a little bit more. So here in the center is a molecule called DNA, and DNA are the instructions for life. And don't worry, I'm going to come back to that. We also have molecules that are called proteins. I like to think of these as the molecules that help carry out these instructions and do all the functions that is needed. There's also a molecule that forms a membrane layer around the DNA, protecting it. And so this is a cartoon of a zoomed in version of a herpes virus. And it actually looks quite similar to what happens if you zoom in really closely on a piece of a human body. So here I'm showing you a cell from a human. We are made of trillions of cells in our body. And it looks actually, on the molecular level, pretty similar to herpes simplex virus. So in the middle, we have DNA. We also have proteins that carry out all of the functions of our cells. And then we have a membrane that provides a barrier, surrounds it, and keeps us all encased together. So just a quick note that although I drew herpes very large in that schematic, herpes is even smaller than a cell, and a cell is, very tiny. So we also have lots of different kinds of cells. So around our mouth, we're composed of skin cells. These protect our inner organs and our muscles and keep a nice barrier between us and the outside world. We also have brain cells, which are shaped a little bit differently. These brain cells help us think, help us feel, help us taste and touch and smell. And we build these cells using instructions called DNA. I told you I would get back to DNA. So usually when we talk about DNA, we think of it as this very futuristic kind of molecule. And as you can tell, I'm a little bit more cheesy, a little bit more old-fashioned, so I like to think of it as a scroll. And this scroll has instructions on it. So for example, these instructions say, make a brain cell. A different piece of DNA could say something like, make a skin cell. Of course, it's a little bit more complicated than that. So here I'm showing you this scroll, and I've kind of expanded it a little bit more. Each of these different pieces of instructions now has a different quotation from a different Shakespeare play on it. On the left, the skin cells are quoting a famous monologue from Romeo and Juliet, and on the right, this brain cell scroll is quoting the to be or not to be monologue from Hamlet, which I thought was appropriate. So I like to think of DNA like I like to think of Shakespeare. It's complicated and it's in code, but once you get to know it and appreciate it, it can be very beautiful. What's different between our DNA and Shakespeare is the fact that although Shakespeare was very productive in his lifetime and he wrote several plays, our cells are even more productive. And the amount of DNA in each one of our cells is equivalent to every single Shakespeare play times 800. So if Shakespeare just wrote the amount of plays he wrote, but 800 more times. Or to put that maybe in a little bit better context, if you were to write all of the information of our cells onto a piece of paper that was a scroll and roll it out, it would stretch 100 miles, which is essentially the distance between Seattle and Canada. So it's a lot of information. And like I said, every cell has all of this information at once. So how does your body know which one to read? So here I'm showing a skin cell. How does it know to be a skin cell? How does the brain know to be a brain cell? And so this is a really interesting trick of DNA. DNA is rolled, and it's rolled around these special proteins that are called histones. And this is really the meat of my research. So here I'm showing two different types of ways that you can roll the DNA. You can roll it really kind of loosely, where you can read a lot of the information on this scroll, or you can roll it really tightly, where the information is kind of cut off and it's rolled up tight. You can put it away and not have to deal with it. And so we use this information to decide what to build. So here in the schematic, I'm showing that we're going to make a skin cell because the skin cell DNA is readable, but the brain cell DNA is not readable. And we store this DNA in a very special compartment of our cells called the nucleus, which I like to think of as a library where there's monks copying and interpreting the DNA and in general taking care of it. So like I said, herpes also uses DNA as instructions. And I'm going to represent herpes DNA in a purple scroll. And so herpes DNA is a little bit shorter than ours, actually a lot bit shorter than ours. It's in fact the length of one Shakespeare play. Pretty much if you took the DNA from one herpes virus, it would be about the length of Romeo and Juliet, essentially. So a lot shorter than our DNA. And herpes also uses our nucleus to read and copy its DNA. So when you get herpes, for example, if you share utensils with someone who has herpes, or you kiss somebody who has herpes, and you get a virus on your skin cells, on your lips, the virus enters your skin, puts its DNA inside, and then uses all of those monks that I was talking about to read and copy its DNA over and over and over and over, and it kind of just hijacks everything. And it does this so that it can produce more of itself, more viruses. And it does this like crazy, and it produces a crazy amount of virus that then bursts out of your cell and kills it. And while it's doing this, your body becomes aware that something's wrong, and your body says, hey, there's a virus, we need to get rid of it. And it sends in the immune system. And the immune system is a defender that tries to protect against all of these kinds of viruses. So herpes needs somewhere to go that it will be safe from our immune system. And it's really clever, and it has chosen to infect our brain cells. And so when you get a cold sore, after the cold sore kind of clears up, the surviving herpes viruses travel into your brain where they deposit their DNA, and they kind of just live there in the sanctuary of our brains for our whole lives. Now, this sounds kind of scary, but I can promise you that herpes is, we don't think it's doing a whole lot in there. It's usually pretty silent, and it's not making more of itself. However, it can be woken up from time to time. And this can happen if you're, say, for example, very stressed, if you get sunburnt, or perhaps sick with another illness. Herpes will wake up. It will start to make more of its DNA. It will start to make more viruses. It will travel down back into your skin cells, and it will cause another cold sore. But this can be problematic from time to time, rarely. This happens because herpes, when we get stressed or sunburned or sick with another illness, it thinks we're dying and it wants to find a new host. So if herpes reactivates to such a large extent, it can be tragic for us. It can cause cell death, brain swelling, and death of the human. So what I'm really interested in is the question of how does the herpes DNA interact with our histones to do what herpes wants to do, which is to make more of itself and to stay alive in our bodies. So we know a little bit about this. Some other labs have shown that herpes DNA will interact with these really kind of open histones when it's actively making more of itself in skin cells to do both make more of its DNA and to package its DNA and escape. However, in the brain cell, it's been shown that typically herpes will hijack those histones that let it roll itself up really tightly and not be read or interpreted. Like I said, it's mostly silent in the brain. But that's not all there is to it. I kind of showed you this very green means go, red means stop histone metaphor, but there's other histones in the body. So the histone that I study is called macro H2A, and I'm just going to refer to it as macro, but if you want, you can call it macromio if that helps you remember, because it fits in with my theme. And so macro is actually a really mysterious histone. Most of the time, macro rolls up the scrolls of our DNA really tightly and prevents it from being read, and that's what it does most of the time. However, in some cells, macro is actually essential to unrolling and reading that DNA. And it's still unclear why it does this or what it can impact. But we do know that macro is involved in some cancers. And so it's important to study macro to understand how it makes the decision to roll it up tight or to open it up and read the DNA. So, what got us interested in this project is the observation that when I infected skin cells in a dish with herpes virus, I saw a difference in the amount of macro in those cells. So, here I took kind of a picture of the macro protein just without any sort of HSV infection. And then I took a picture of the protein over 15 hours of infection, and you can see that the macro protein levels have gone up steadily over time, which is really interesting. This is the first time that macro has ever been described to be involved in any kind of virus infection. And the question is, why does macro matter for herpes? Most of the time it's keeping DNA really tightly rolled up. You wouldn't think that something that wants to be very active is going to use this protein to make more virus. So I wanted to ask, what happens to herpes if macro is gone? So I had some cells that have macro. I also made some cells that don't have any macro. And then I infected them with herpes simplex virus. And then I harvested the virus that came off. So here I'm representing that by a pink little liquid or a gray liquid. And I diluted this liquid over and over onto another cell line. And so what's important about this is that you can actually measure the amount of virus being produced because you can see this purple here is now representing the cells. You can see these white zones of death that I'm representing here with a white dot. And that's what happens when one virus gets into one cell and spreads over time and kills its neighbors. And you can actually count that. So for example, cells that have macro, you would expect quite a lot of virus to be produced off of them. These are just your normal cells, and herpes replicates quite well in them. However, if macro was important for herpes, you would imagine that there would be fewer actual viruses produced by these cells that don't have macro. And in fact, that is exactly what we see. So here I'm showing you a picture of this assay that I actually ran. Each little zone of death here is where one virus got into a cell and spread to its neighbors. And so you can actually count how many viruses there were produced by these cells, and you can quantify them. And so I'll show you this here. So in cells that have macro, they produce about 200,000 viruses per milliliter after 12 hours of being infected with herpes. However, cells that don't have macro actually produce 10 times fewer, 20,000 viruses, which is quite a significant reduction, I think. And so my conclusion is that macro is somehow helping herpes make more virus. So here I'm showing that macro is perhaps helping herpes make more DNA or helping the virus escape the cell. But when you don't have macro, this scroll can't stay open as well, kind of rolls back in on itself. And it's not able to read this DNA quite as well. And so you get fewer viruses. So I have a lot of open questions that I'm not going to get to today. And so I'm going to end on all of these questions. So the first is, how and why does macro actually help herpes? At what stage does this happen? Does it happen at the stage of making the DNA, or maybe it happens at helping the virus escape the cell? And an important question is, how does herpes actually convince macro to help? It's kind of weird that our own histone is helping a virus make more of itself and kill our cells. I'm also really interested in the question of what happens in the brain. Does macro do what we normally think it does and keep herpes kind of silent and tightly rolled up? Or is it acting weirdly and somehow helping herpes make more virus and wake up, even if that means that we're going to get sick or possibly die? So like, unfortunately, unlike my introductory sentence, I don't have the answer for you in this story. We're really only at the beginning of this story. So if this was a Shakespearean comedy, Mac Romeo and Herpes could live happily forever after in the brain of our cells and keep herpes quiet, and then everyone would be happy. But if it was a Shakespearean tragedy, macro will help herpes wake up and make more of itself and maybe cause sickness. And so I'm working on it, but the big answer is how can we use this knowledge to prevent this tragic ending? I mean, I think all of us kind of secretly wish that Romeo and Juliet didn't end tragically and was actually a happy ending. So that's kind of what I'm hoping for my story too. And I'd really like to thank my lab for helping me with my research and also helping support my dramatic flair. Here we are wearing a bunch of sparkles on our Sparkle Spirit Day. I'd like to thank the program in molecular and cellular biology at the University of Washington, the Fred Hutch Cancer Research Center where I work, and also the Engage class for such a great quarter. And then I will take questions.
Great. All right. Thank you, Hannah. Christine, I'll invite you back on and we'll go through some questions. Thank you all. Thanks to everybody who submitted questions so far. We're going to start with questions for Christine. So the first one is, does the ever-shifting sand along a coastal area mean rip current locations can also shift locations or intensity?
That's a great question, and it totally can, because it would shift the first example of the type of rip current I talked about. So that's the channel rip current. And so, for example, after large storms or even moderate level storms on the East Coast, that's when channel rips can be the worst, because the sand is shifting along the coast and it's actually making more areas where channel rip currents can form. So that's something that people are very interested in, and people actually try to get better estimates of how much sand is, or the seafloor shape is evolving over time.
Okay, next one is, what was the success rate of creating a rip current in your experience? Did it take several attempts to create one, or were you able to make them on command?
So luckily, actually, my advisors had gone to this tank before we ran our full lab experiments, and they'd seen that they could actually make these flash rip currents. Otherwise, we wouldn't be completely sure. It would be like a gamble if we would go there and be like, well, we're making these waves. Are we actually seeing the flash rip currents? And so during the experiments themselves, it's not like we could make them on command. We knew that certain types of waves, during certain types of waves, we would see more of the flash rip currents. But the timing of them is something I haven't resolved, if that's how that relates to when they occur. So no, we couldn't completely predict it.
That was a really impressive lab. Pretty fun to see. So the next one is, could the fact that the tank contains fresh water impact the applicability of your results to rip currents in the field? Might salinity have an impact on transient rip current formation?
Yeah, this is a question I get quite often, because if you're doing smaller scale experiments even than the one that I'm doing, then the difference in the density of the water, so salt versus fresh water, might impact the fluid motion. But the fluid motion that we're studying on the scale we're studying it, we don't really think it has a large impact on the formation of these rip currents and why they form, which is what I'm trying to answer.
And then the last one, obviously we can't outfit every beach with stereo camera setups. So what is the minimum amount or type of information or measurements we need to make a difference in flash rip current forecasts? I think basically maybe what's the minimum amount of information and collecting information technology that we need?
Yeah, some people are, instead of putting out multiple cameras, they're putting out just a single camera, and you can actually take images throughout time just with the camera, and you can average the images over time, and so that you don't see the wave crest, but that you can see the rip currents that occur. So some people are doing that, and I think that would probably be the least expensive way to actually start to take measurements of these flash rip currents.
Cool. And then I got one last one that just came in. Is there a typical time scale over which flash currents occur?
Yeah, so they're much faster than the channel rip currents, as I mentioned during my talk. And they can occur anywhere from like on the order of like 30 seconds to much longer, maybe on the order of like 5 minutes or even longer for really large ones. So they're much shorter than the channel rip currents. It can last for like, it can be constant given a set of waves. But yeah, on the order of like 30 seconds to 5 minutes or 15.
All right, well, thanks so much for your presentation. It's great. Okay, so Hannah, what is your opinion on the categorization or lack thereof of viruses as living organisms?
That is a can of worms. I'm personally okay with it. I think it's kind of a nitty-gritty difference, and I think some people can be kind of mean about it. If you say, "Oh, that virus is alive," and they'll say, "No, it's not alive. Viruses can't be alive." But I think biologically the difference is solid. You know, viruses quite literally can't do so many of the things that, let's say, bacteria can do. So like there's just a lot of essential components that they don't make, which makes me believe like, yeah, they're not like truly alive. But don't be mean.
Cool. Does herpes wake up from stress or sunburn or sickness because our immune system is compromised and it is just taking advantage of that?
So that's a great question. So immune system problems are another reason that herpes can wake up, is if your immune system becomes compromised. The actual reason that it wakes up due to stress is actually due to the way that your brain cells are talking to each other. So I think that one has less to do with the immune system, although I believe sunburn probably could have a little bit to do with the immune system. Say like if you got sunburned on your mouth and your immune system is right there, that could help herpes wake up. And I bet that being infected or having some other kind of disease would definitely involve the immune system.
How do individual DNA histones know to be loosely or tightly wrapped?
Yeah, that's a great question. I kind of hinted at this in my presentation. So the histones themselves are those like rod-like cartoons that I made, but you can actually add different chemicals onto those rods. So I kind of hinted at that when I had a flag or a stop sign. And so that's a way that these histones can be changed to either be more tightly or more loosely wrapped. It kind of depends on the chemicals that are put onto the histone. And that's a whole field of biology unto itself. It's also really fascinating and really cool.
Is it known when macro evolved? Could macro and herpes have evolved at the same time in the human timeline?
Yeah, that's a really excellent question. I have done a little bit of looking into that. Macro, I will say that macro is in all animals that have a spine. So not all vertebrates, not just mammals, not just humans. And I don't think that macro and herpes evolved at the same time, because herpes simplex 1 is very specific to just humans. Like it's just us that it infects. So we have macro, like chickens have macro, platypi have macro, but we are the only people who get herpes. But the coevolution question is a really excellent one that I'm interested in.
Great. Well, I want to invite Christine back on and just thank you both so much for presenting with us tonight. These are super interesting topics, and I hope that your research in them continues to go well. What year are both of you in? Are you both near the end? of your work?
I'm in my 4th year, so I've got a few years to go, but on the middle end-ish.
I'm also on my 4th year, so on a similar page as Hannah.
Super, great. Well, congratulations to you both. Thank you again for presenting with us. And I want to thank the audience as well. Thank you all so much for your questions. And you can check out more of what is coming up at Town Hall on our online calendar. The next UW Engage Science program is next week, Monday, the 26th. So hopefully we'll see some of you there as well. So thanks again and have a great night.